Dr. Hara Prasad is a physician and resident doctor at the University of Delhi, India, and a computational medicine researcher at Ashoka University. He has worked on various projects involving AI and ML healthcare with the Government of India, MNCs, and health tech startups over the last 2 years. He completed his MBBS from the University of Delhi and his PG MLE from NLSIU Bangalore. Dr. Mishra specializes in clinical pharmacology, digital health, computational medicine, and integrative medicine. His research focuses on bridging current medical therapies with computation for next generation medicine. He has also authorized India's first medicine documentary on COVID-19 by a medical physician. His vision is to touch a billion lives. Having said that, I am handing over the virtual mic to Dr. Hara Prasad Mishra, who will be presenting today on genomic surveillance of COVID-19 variants with language models and machine learning. Over to you, Dr. Prasad.
Thanks, Yamina, and all the HITLAB organizers for having me here. Pleasure to be here and speaking today. To start with, I will be speaking on the topic that is genomic surveillance of COVID-19 variants with language models and machine learning. To give a brief perspective, back in 2020, when COVID struck India and a lot of haywire happened and we are into the pandemic, the government of India came up with the idea that the PSA office, that is the principal scientific advisor to the PM, came up with the idea that we need to consolidate the scientific infrastructure of India. And as a result of which, Delhi Science and Technology Cluster you can say got a birth, and all the big institutes, be it medical, scientific, R&D, some startups and some MNCs came together. And one of the birth childs of this big consortium was this particular project, which I will be presenting, and how it has revolutionized the pandemic preparedness for India. And a lot of international countries have started taking inspiration from it. To start with, these are my fellow colleagues who have worked with me for this project, and 4 institutes we are representing particularly. So to give a brief understanding exactly what this whole project and whole session is all about, that we know that predicting the case load and also getting to know about the upcoming variants of COVID-19 is a challenge. And also it's very important to plan any public health intervention. So for this, we present you strain flow, which is a supervised and causally predictive model, which uses unsupervised latent space features of SARS-CoV-2 genome sequences. The strain flow model that I will be presenting now, it was trained and validated on 0.9 million sequences for the period of December 2019 to June 2021. And the model was then frozen up, and we prospectively validated it from July 2021 to December 2021 with a sequence of 1.9 million. Moving further, this strain flow captured the rise of cases, particularly in terms of whether it is going to be an uptake or a downtake, or there is going to be a plateau in the number of cases 2 months ahead of time. Especially to our astonishment, even the Delta and Omicron surges in most of the countries, including that was in India, was predicted. And for Omicron, we are spot on. We predicted it in the beginning of November 2021. I will be showing you, we did something called entropy analysis, which was done both in a quantitative as well as a qualitative intervention of the strain flow, unsupervised embeddings, which clearly revealed that explore-exploit cycles in genomic feature space could add up interpretability to this particular kind of deep learning-based model. We also conducted a codon level analysis of our model for interpretability of these results, which I'll be showing now, and biologically validated them for the unsupervised settings. Now, starting with the heads up, that is how the architecture of this whole strain flow was built up. So if you see, there are 6, you can say, a set of things I will be presenting. The first is talking about that COVID-19 genomic sequences we selected from GSED. And if you see, we selected 0.9 million sequences. So these were actually selected from GSED EpiCov database. These are very high quality sequences with complete temporal information, and it was then further filtered. What we did as the next step was that we extracted the spike gene region of these sequences from FASTA files using COV2 sequence tools. A CSV containing these sequences and other metadata such as country names and dates were further created. In the third, you can say step, or in this particular thing, the skip gram, which I'm showing as the second, that is the language modeling with word to vec, we simply did is that the sequences were splitted into chunks of 3 characters, which we call in biology as codons. Uh, these were splitted sequences which represent a document, a 3-letter word. That's why we call it a word to vec model. And we trained further this word to vec model on the spike gene sequence for learning the 36-dimensional word embedding. The average of all these word embeddings in a given sequence was further treated as the embedding of this sequence. We calculated the sample entropies, just like you can see in third and fourth. The latent space analysis, both qualitative and quantitative, was done, and we did the sample entropy calculation for each dimension and for each country and for each month, which we'll be discussing further. After that, what we did is that we needed new COVID-19 cases for each country, for each month, and these were calculated using the database from Johns Hopkins University repository. Then, as the next step, what we did is that a feature selection algorithm was built, which is called Baruta algorithm, and it was used for selecting the entropy dimensions for prediction of case loads 2 months in advance. And as a last step intervention, what we did is that we built a random forest regression algorithm that was used for predicting the new cases 2 months ahead of time. These inputs to the models are the country names, the important features that we extracted from the Baruta algorithm. The predictor variable in the case load were always 2 months ahead of time. And this was the whole architecture that I was describing. This is the latent space change analysis. And if you try to see for B, C, D are the T-SNE plots, whereas the E is the quantitative, you can say, latent space analysis. And we are comparing different countries and on a different timelines. So this was a very good capture for us. The training period was this, whereas after that, there was a prediction and there was a testing phase. Now comes the questions of what is the uniqueness of research. A lot of research in the last 2 years happened in this space. They talked about how we can predict the increase in the number of cases. Some even predicted the exact case loads, and they were based on a lot of modeling. Practically speaking, ours was one of unique and first time in the whole globe in which we treated codons in the sequence as words in a document. Simply, that's why we call it a word-to-vec modeling. We trained language models to understand the underlying grammar of the sequence, breaking them down into 3 documents, and optimize them the number of latent dimensions in the grammar space. Just like I mentioned, a 36-dimensional vector space represented the optimized grammar. Towards the late end, we did the sample entropy. Entropy means the noisiness, the haziness in the data. And these dimensions represented the mutation, because as you know, the mutation is also like a noise in a sequence. Similarly goes with the entropy. And we devised this machine learning captured dimensions of choice, that is the dimension of concern, DOCs, what we'll be calling now, for subsequent months. And these were validated biologically because in our team, doctors were also there, physicians like me, and we validated them whether transmissibility was getting affected, how the vaccinations was getting affected, how the infectivity rate got affected and changed due to this VOCs, that is variant of concerns. Now comes the next thing, that while devising this model, we believe that we need to follow some core ideas. The core ideas were 4 principles we followed. First is that the virus DNA sequences can be treated as documents with words, that is the codons. The word pattern changes, that is the mutations as per biology. These are leading to change in the entropy, that is the noisiness in the underlying space. Just like I mentioned before, we extracted special dimensions, DOCs, that predict spread using machine learning. And DOCs provide a practical approach to define this. If I talk about prospective validation, this is the prospective validation we did, and it is 2 months ahead of time. That is, we predicted the case and simultaneously the case loads as per the JHU and the prediction. So JHU is a Johns Hopkins University. If you see the red line is showing the caseload that was as per prediction, as per the actual, you can say, JHU database. And blue line is regarding our prediction, you can say what we exactly calculated. And I completely agree. We missed on the part that we could not predict the exact magnitude of the cases. But if you try to understand the upsurge or the downsurge, if there is an uptick or there is a downtick, we were spot on that part. Even to a great extent, we predicted a lot of plateauing as well. So that also was spot on. If I talk about the dimensions of concerns and that how we did the predictive analysis to understand it, we need to understand one thing that we selected 36 dimensions, and out of these 36 dimensions, we could find that some dimensions were very relevant, and this is the, you can say, heat map. And in this heat map, we could figure it out that despite the fact that our model cannot directly capture the SARS-CoV-2 variants, it was observed that dimension 32 captured the CTG, CGG, which were rank 5 and rank 8 respectively. And these were known to be involved in the mutations, which are very prominent, like T19R. Similarly, if we talk about dimension 3, it captured 3 codons, that is ACG, CGG, and CAC, that were associated with multiple variants such as K417T and L425R, which caused increased infectivity, pathogenicity, and spread in COVID-19. If I talk about dimension 30, it also captured codons like CAT and CAC in the heat map, and it got associated with D1118H and Delta 69, which were a part of B1.17 lineage. If I talk about dimension 32, it captured multiple codons that were linked to B.1.429 lineage, which was a part of L452R mutation, a very prominent mutation causing havoc throughout the pandemic. And dimension 3 captured lineage associated with the B.1.617.1. And this was one of the first observed in India, if I talk about B.1.617.1, and in December 2020. And we were one of the pioneers in spotting it. And we got appreciations from Government of India, and also featured in CDC Hot News for this, because we predicted that it will be having increased infectivity and transmissibility. If I talk about other figures, or particularly the graphs I'm going to show for England, India, and USA, you need to understand that we are talking about the entropy dimensions. So what we did exactly is that we selected dimensions which were linked to Delta variant, and these dimensions included dimension 3, 4, 12, 13, 15, 16, 25, 28, 30, and 32. These are few of the dimensions we selected out of those 36 dimensions. And our case prediction model, as I mentioned earlier, was frozen in June 2021, and prospectively predictions were done for the case loads from July 2021 to December 2021. Our model predicted the case upsurge in India due to Omicron variant, just like I mentioned earlier, in November and December 2021. And it was 2 months ahead of time. And I will just say it again, it was spot on. And we really appreciate the model. And also we got even astonished that how the model was so spot on, especially for the Omicron thing. Although the model failed to predict the exact values of cases, just like I mentioned, the magnitude, the amount of magnitude we missed to certain, but yeah, it was a useful trend indicator in terms of whether there will be an upsurge or a downsurge. Further, we observed the explore-exploit cycles in the entropy space of India prior to the case peak due to Delta variant in May 2021. A similar exploration phase can be observed for the months from September to November 2021, which is talking about the upsurge due to Omicron in the upcoming months. Before moving and concluding my session, I want to just mention that our genomic sequence-based language modeling that I was presenting was for COVID-19, and it was limited to spike gene aspect. We tried to preserve the spatiotemporal information of SARS-CoV-2 and tried to depict it with a phylogenetic analysis, which tried to capture the geographical outlook, that how different countries, how different variants were behaving during the pandemic. And through entropy, which is the noisiness, we tried to capture the latent space dimensions, variability in the spike gene, and simultaneously use these entropy dimensions that could be predictive of new COVID case loads. And towards the end, we also simultaneously validated all these things biologically, that is whether the upsurge that is going to be, whether the variants that are upcoming, whether they can be variant of intent or they can be variant of concerns, and how they're going to affect the transmissibility part, the infectivity part, the spread, and all those things. Coming to the limitations, because we all practice science, and science is always evidence-based, and no research is complete without limitations, to be very frank. If you can say researcher says that there is no such limitation in our model, then I am not going to accept it. Every model has got its own limitation, same goes ours. I will be discussing few of our limitations. The number one is that our approach of selecting codon embeddings does not indicate the positions where the codon change may have happened in the spike gene. This was exactly because the low dimensional embeddings we created do not preserve the positional encoding of words. However, we are currently investigating and working on advanced approaches such as complex valued word embeddings, which can capture positional encodings, and transformer models such as BERT, which I'm currently working on to overcome our current limitations. The second limitations that I could think of is that our strain flow model is trained only on the spike gene data. The spike gene data of the viral genome, so it does not exactly represents the complete variation spectrum of the virus as such. And to mitigate this shortcoming, we will develop a genome level strain flow pipeline in near future for SARS-CoV-2 particularly. And further, the third limitation is that the present study is not considering the interaction between spike gene and other genes in the SARS-CoV-2 genome as such. So we have not particularly considered the interaction between H2 receptor sequence for humans and spike gene sequences as such. The reason was very simple. We could not get easy availability of such large-scale paired data. And fourth, the limitations is that our current approach does not explicitly capture any sort of specific positional mutations that I could think of immediately. And finally, I will say that the fifth limitations, or you can say the promising thing that we need to work computationally, is that our model was trained on a relatively small number of samples, and we used a very small number of samples to construct the supervised predictive model for case prediction, which is only 0.9 million sequences. As more data becomes available in subsequent months, we can definitely produce more confident case predictions models. Particularly speaking, an empirical validation depicted that we require a minimum of 100 samples per month for calculating this sample entropy, that is a noisiness I talked about earlier. This also reminds me and underscores me that there is a hard need for a more reliable and agile approach to deposit country-level data sets of genomic data on repositories such as GSET. And I take this platform to appeal to all the countries and all the, you can say, big leaders to facilitate the sharing of such genomic data in order to be prepared for any future waves of current pandemic and for preventing the new emergence of strains. I also believe that this kind of study is an instance of the new paradigm shift we are going to have in pathogen surveillance using a novel language modeling approach. And this is potentially scalable to infectious disease surveillance and in near coming days as we move apart to antimicrobial resistance as well. So these are my references. The first one is our paper, which has got featured and got published in an international journal called Frontiers in Genetics back in April 2022. And second is our dashboard. You can refer to it. It is for the public use, and you can see how we are predicting the caseloads and how predictions are there. This particular research got featured both nationally and internationally, but I want to feature this particular news coverage from the Hindu, in which in the science section, we have covered that how this technology is going to define the new age pathogen surveillance for India and for the international world. This is my team and my mentors, Dr. Tapritesh and Dr. Rintukutum. And these two guys have inspired me a lot from shifting my gears from mere a physician to a clinical researcher and simultaneously working in the data science and machine learning space. And for the last 2 years, I'm working in that space. I'm also part of Center of Excellence of Healthcare at IIIT Delhi, and this is our lab at Tab Lab. And in the right side, you can see my team. So yeah, that's it in my presentation, and open to queries and happy to answer them.